well for a long time. And although Arya will talk about a lot of Svati's scientific accomplishments, I'll mention very briefly that she's absolutely tireless advocate for education, for mentoring physician scientists, particularly women who are interested in pursuing a scientific career. And last, and probably least, she actually was my very first day of cardiology fellowship in the Echo Lab. So with that, I'll turn over the podium to Arya.
It is a great pleasure actually to introduce Dr. Svati Shah, Professor of Medicine, Associate Dean of Genomics, and Director of Duke Precision Genomics Laboratory at Duke University Medical Center. Dr. Shah received her bachelor degree at Johns Hopkins University in biology, and then did a Master of Epidemiology at Johns Hopkins, and then continued to do medicine at the University of Washington School of Medicine in Seattle. Then she went to Brigham and Women and did her internship residency, and then following that, she went to Duke to do her cardiology fellowship and train in genomics, medical genomics. Her translational laboratory studies genetics and metabolic pathways in a variety of cardiovascular diseases. Her recent focus has been in heart failure and HIV, diastolic dysfunction. She has been involved in biomarker discovery and has been actually, for that work, a PI of the Google project baseline at Duke. Her clinical interest actually overlaps with mine also, and she has interest on monogenic cardiovascular diseases. She has been a recipient of many, many awards. She's a fellow of American Heart Association and Association of American Physicians. She's editor of many journals, including Circulation. Her clinical interest is, as I said, is mainly monogenic diseases, cardiovascular disease genetics, as well as common and complex disorders, early onset CAD, hereditary aortopathy, familial cardiomyopathy, etc. She has been recipient of many NIH research awards, has co-authored more than 150 publications, some of them actually with our current chief, Dr. Velasquez. Apparently they are very good collaborators. And I don't want to blabber and let her to talk. Her topic of talk is tackling the obesity epidemics. One size does not fit all. Dr. Shah, please.
Thank you so much, Dr. Mani and Dr. Friedman, for the wonderful introduction. I'm blushing for those of you who are able to see me. It's such an honor to be able to present to the Yale faculty. I wish I could be there in person. I've actually never visited Yale, and I was very much looking forward to physically seeing all of you. I have very many good friends at Yale, and very many people that used to be at Duke that we miss a lot. Eric Velasquez, who is chief of cardiology, obviously, who I see there in the background, who we miss dearly. Eric used to be my Echo Lab work buddy, so we've collaborated many hours together. So it's really an honor to be able to present at Yale. Let me just get this going while I am able. And I know everybody's Elizabeth, you let me know if my slides aren't working. So I know everybody's days, and I would say even my nights, because I dream about COVID-19. Everybody's thoughts are occupied with coronavirus. But I thought we could take a brief interlude to remind you about an even bigger public health epidemic, and that's the public health epidemic of obesity. And while you guys might be rolling your eyes, and you guys are not on video, so I can't see you rolling your eyes, you might be saying, well, we're cardiologists, why do we care about obesity? Hopefully what I will be able to tell you over the next hour is not only why we as cardiologists need to be very aware of the clinical implications of obesity, but also of the biologic links between obesity, cardiometabolic risk factors, and cardiovascular disease. And so what I thought I would do today is to really outline obesity as the true public health epidemic that it is. I am a molecular epidemiologist. My lab really focuses on understanding the molecular biomarkers, molecular pathways, and how those biomarkers might be used in better risk stratifying, understanding the molecular underpinnings of different diseases, with obesity being one of my recent passions because of its public health implications. So we'll talk a little bit about the public health implications of obesity, talk a little bit about the molecular insights that my lab and other labs have into the link between obesity and cardiometabolic disease, talk about how some of this work has led to biomarkers which will give us hopefully more personalized approaches to obesity management. And then, yeah, I mean, I love biomarkers, fine, but we do need to talk about more than just biomarkers when we think about obesity intervention. So we'll talk a little bit about work that's being done across the country that is really outside my sphere of expertise, but I find very provocative as we think about the huge impact that we can have on cardiovascular disease through interventions on obesity. And then finally, what do we really need to think about as we think about the short-term and long-term future to solve the obesity epidemic? I just wanted to say what this talk won't be. This talk is not going to be a talk about all the different ducks that are around there. Those of you who spend time on Twitter know there's lots of heated discussions about keto diets and plant-based diets, and even people who are voraciously against whether you should even eat an avocado. This is not going to be a talk about that. We're not going to talk about different kinds of diets. So let's talk about obesity. Obesity is a true public health epidemic. And again, while we are all mentally and many of us physically obsessed with coronavirus, once all this has died down, we're going to be left with the physical consequences of people staying at home, as well as what was already the foundation of a growing epidemic of obesity. So unfortunately, we in the South are afflicted with the epidemic of obesity more than you guys in the North are. And in North Carolina, this is a really surprising epidemic. When I moved from Boston to North Carolina 19 years ago, even back then, it was a surprising epidemic, and it's only grown over the past 2 decades. Excuse me. So over these are data from a couple of years ago. And it's really surprising. 65% of adults in North Carolina are either overweight or obese. And the projections currently are that by 2030, 58% of North Carolina adults will be obese. So not even just overweight, but will be obese. And when you look at the heat map across the United States, you can see that while the South is predominantly afflicted, you can see that even states that we think of as very healthy, such as the Northwest, which is where I grew up, that even those states have a surprising amount of overweight and obesity. And then while we might think of this as a problem of developed countries, this is in fact a problem not just of developed countries, but also a problem of developing countries. And it really is becoming an international epidemic. So you can see across the world heat map that even European countries and countries that we don't normally think of as being afflicted with obesity are becoming increasingly afflicted with obesity. Of course, when you look at some parts of the map, you might think, well, we think of India as having an epidemic of diabetes and other cardiometabolic diseases, but they don't look like they have an epidemic of obesity when you look at this world map. But in fact, a lot of that may be because of the way that obesity is defined, and some of those metrics are not appropriate for different ethnic groups. So while you guys in Connecticut are in good shape, literally and figuratively, you're one of the least obese states in the country. I would urge that it is not just a problem just while you guys are the least obese. You are still not in the clear. You still have worrisome obesity rates. So in Connecticut, you can see here on the right-hand side of the screen, obesity rates by age from 2018, and summarized on the left-hand side of the screen, 27% of adults in Connecticut have obesity, and I think these numbers are even more worrisome just when you look across the country. 29% of Connecticut children aged 5 to 17 are either overweight or obese. 11.5% of youth ages 10 to 17 have obesity, and almost 13% of high school students have obesity, not even an overweight, just obesity. And when you look at the map on the bottom right-hand side of the screen, you can see that, again, while you guys are one of the least obese states in the country, your obesity rates continue to increase as the obesity rates across the country continue to increase, at least adult and adolescent obesity. So why should we care about obesity? We're cardiologists. Many of us are cardiologists. Well, we know that there are many downstream consequences of obesity. So, you know, this is a nice graph from a paper that really outlines some of the clinical diseases that are linked to obesity. Pulmonary
We lost your voice.
Okay, are you guys able to hear me again? Sorry about that.
Yes, yes, now.
Okay, I am so sorry. So I will just go back to this slide, just reminding all of you that, that the obesity is not just a disease in and of itself that we don't need to worry about as cardiologists, but there are significant downstream consequences of obesity. So every 5-unit increase in BMI is associated with a 29% increase in overall mortality, a 41% increase in vascular-related mortality, and a 210% increase in diabetes-related mortality. And in fact, many of us are interested in heart failure, as my lab is, and 11% of heart failure in men can be attributed to obesity, and 14% of heart failure in women is attributed to obesity. And in fact, this has prompted the American Association of Endocrinology to propose that we shouldn't just call it obesity, but that we should call it ABCD. Can you guys still hear me?
Svati, we cannot see the
slides.
Sorry, guys. Better?
Yes, we see it now.
Okay. You didn't miss anything. You missed some of the alphabet. Sorry about that. So this has really led the American Association of Endocrinology to propose a new term called ABCD. So to not just call it obesity, but to call it adiposity-based chronic disease, to really reflect the fact that it's not just obesity, but all the downstream consequences of obesity. Elizabeth, thank you for the texts. Those are super helpful. But as we sort of transition into some of the molecular work that's been done, I think it's really important to point out that there is marked heterogeneity in the development of the metabolic and cardiovascular consequences of obesity. So, for example, when you look at the left-hand side of the screen, these are data from NHANES. This is sort of stratifying people who have normal weight, overweight, and obesity into clinically determined metabolic health or metabolically abnormal metabolically abnormal criteria. And you can see that, as you would expect, people who are of normal weight are more likely to be metabolically healthy, and people who are obese are more likely to be metabolically unhealthy. At the top is men and the bottom is women. But in fact, there's marked heterogeneity in there, where you have people who are of normal weight who are metabolically unhealthy, so 30% are actually metabolically unhealthy. But when you look at obese individuals, and this is in the top screen looking at men, that 29% of obese individuals are actually metabolically healthy when you consider them from a clinical standpoint. And concomitantly, looking at the right-hand side of the screen, when you look, these are data looking at events. So this is looking at hard cardiovascular outcomes in patients stratified by body mass index, and you don't need to look at all the hazard ratios. But when you look at people who are overweight and obese, you find that there is marked heterogeneity. When you look at these hazard ratios across the BMIs, there is a lot of heterogeneity of what the risk of having a cardiovascular or all-cause mortality event is if you're overweight and obese. And in fact, most people who are lean, when in fact many of them may be at risk of cardiovascular disease because of their metabolic unhealth. So I would propose, and I think many people would propose, that we need a more personalized approach to obesity management. And this would take into account many different components of both environmental factors, the things that we know that contribute to obesity from the way that we live. We're going to talk a little bit about this. This is not work that I do, but thinking about urban planning and things that really are part of our infrastructure that determine how well we are able to live in our world and be healthy. Things like psychosocial factors, and then obviously work that my lab is interested in, thinking about genetics and molecular that might determine. Paper that came out, and slightly came about six to 8 months ago, thinking about the genetics of obesity, right? So we sort of think about, well, maybe there's a genetic underpinning of obesity, and maybe this genetics really determines the heterogeneity of not only whether you develop obesity, but if you have overweight or obesity, whether you have a cardiovascular event. And I think what's really interesting about this paper is that it, for those of you who looked at this, they used a polygenic risk score. So, you sort of think about, oh, maybe it was 1 or 2 genetic risk factors, but in fact, these were up to millions of genetic risk factors that were combined together, that only when you combine these lots and lots and lots of genetic factors together were you able to explain obesity, and also able to explain the downstream risk of different downstream risk factors or downstream consequences of obesity with these genetic risk factors. And it still didn't explain the large amount of cardiovascular and other complications of obesity. So sort of taking a step back, and this happened in my career when I thought about a lot of different components of cardiovascular disease. I'm trained, in genetics, and I have a strong background in genetics, and I love genetics. I take care of genetics patients, as Dr. Mani and many of you do. But when we think about these common complex diseases, it's kind of a little bit strange to think about telling, for example, a baby what their risk of having a cardiovascular event will be when they're 50, 60 years old, when we know that there's so many things that happen to that child and adolescent over the course of their lifetime. And we know that that static DNA variation changes over time. And so, like many of you who do translational research, what my lab has really been doing over the past decade has been to say, okay, well, yeah, absolutely, static DNA variation is important, but looking at how that static DNA variation changes and is expressed differently over time. So expression when we look at RNA, but also proteins, and then what my lab has really been focused on over the past decade is looking at metabolites as sort of these downstream byproducts and integrated reporters of this sort of static DNA variation, but also how that static DNA variation integrates with environmental factors and changes over time to create the end phenotype. And I'm lucky enough to have my lab within the Duke Molecular Physiology Institute, where we have basic scientists, translational scientists, as well as core labs to do all this integrated omics science. So again, sort of focusing, really thinking about this heterogeneity. How do we dissect out this heterogeneity so that we can have clinically important findings that might help us take better care of patients, as well as dissecting out the biology, the sort of molecular insights of this heterogeneity. And so for the next few minutes, I'd like to talk to you a little bit about work that my lab and other labs have done that have hopefully given us a little bit of a molecular insight into obesity and its relationship to cardiometabolic disease. So this story sort of started almost a decade ago in work that I did with Chris Newgard, who is a biochemist who is the director of the Duke Molecular Physiology Institute, and he is a very close collaborator of mine. And we asked a simple question in this study, the Stedman study. We asked a simple question using metabolomic profiling, using a targeted metabolomic platform. We asked the question of, can we distinguish obese individuals from lean individuals using these circulating metabolites? And in fact, what we found was there was a cluster of metabolites, branched chain amino acids, as well as the mitochondrial catabolic byproducts of branched chain amino acid catabolism, these C3, C4, and C5 acylcarnitines on the right-hand side of the screen. They all clustered together statistically in a principal components analysis factor, factor 1. And this factor 1, these levels of this factor 1 were higher in patients who had obesity compared to patients who were lean. And again, I can't see your faces, most of your faces, but you guys are hopefully rolling your eyes and saying, well, did you really need to spend hundreds of thousands of dollars doing metabolomic profiling to identify metabolites that discriminated obese from lean individuals when we could have done that just by looking at those individuals? But in fact, what we found was not only did these metabolites distinguish obese from lean individuals, but that these metabolites correlated with measures of insulin resistance, and that they correlated with HOMA, not just in obese individuals, which are in the open squares, but correlated with HOMA also in lean individuals in the dark triangles. And so this sort of mitochondrial cartoon on the right-hand side of the screen demonstrates, and in bold, we have all the metabolites that clustered together and really showed us that this excess branched chain amino acid and increased branched chain amino acid mitochondrial catabolism seem to be at play in obesity and insulin resistance in this study. So Chris went back to his basic science lab and showed through a feeding study where he supplemented rats. So in blue, we have standard chow, and in yellow, sorry, I'm trying to use my pointer. In blue, we had standard chow. In yellow, we had high fat. And in pink, we have high fat supplemented with branched chain amino acids. That, in fact, when you supplement these mice with branched chain amino acids, even though they actually gained less weight than the high fat fed rats, they actually became equally insulin resistant as the high fat fed rats. And this was demonstrated by these glucose tolerance studies, as well as insulin stimulated AKT in the muscle and insulin levels, suggesting not only that these were markers of insulin resistance, but that in fact branched chain amino acids may in fact be etiologic in the obesity and insulin resistance process. And so thinking more about what are these branched chain amino acids doing that are so toxic, this is work that we did with Yibin Wang's lab. And while this is a heart failure model, it does start to tell us a little bit about the biology of how branched chain amino acids may be toxic. And there's been a lot of work done over the past 10 years in the biology, but I think this is sort of an interesting study. And what Yibin's lab did in this case, again, we were collaborators on this. So this is a pressure overload model of murine heart failure using TAC. And what we found was coordinated regulation of the branched chain amino acid catabolic enzymes in the pressure overload model in these mice. And interestingly, when we find this downregulation of branched chain amino acid related catabolic enzymes, it sort of mimics the fetal mouse hearts. And in parallel, we found higher branch chain keto acids. So branch chain keto acids, that's byproducts of this catabolic defect in branch chain amino acid catabolism. And then in fact, it was these branch chain keto acids which appeared to actually cause an inhibition of the respiratory chain, inhibited superoxide production in isolated mitochondria. So not just that the branch chain amino acids were the problem, but it was because of branch chain amino acid catabolic defects that led to excess branch chain keto acids, and that it was actually the branch chain keto acids that were toxic. And when Yibin's lab gave pharmacologic enhancement of the key enzyme branch chain keto acid dehydrogenase, that in fact it blunted cardiac dysfunction, because when you enhance branch chain keto acid dehydrogenase, it relieves that block in branch chain amino acid catabolism, and you have reduced branch chain keto acid. So suggesting a biologic mechanism whereby another biologic mechanism whereby these amino acids may be and get your branch chain amino acid levels. And in fact, there's a lot of things that determine what your branch chain amino acid levels are. Those tissues, so the amount of adipose tissue you have and where your adipose tissue is partially determines that. We know the gut microbiome and what kind of microbiome you have determines your branch chain amino acid levels. What you eat, essential amino acids, branch chain amino acids are essential amino acids. Genetics actually plays a pretty small role in determining your circulating branch chain amino acids. And then I'm going to tell you a little bit about how the liver may play a role in your branch chain amino acid levels. I wanted to tell you just really quickly about some work of the role in the microbiome in branch chain amino acids. This is work from Jeff Gordon's lab in collaboration with Chris Newgard that came out several years ago, led by Vanessa Radura, who's actually at Google now and working with us on the Google baseline study. This was a study where they did fecal transplants. So they took humans who were discordant. These were twins. So they took 4 twin pairs. The twins were discordant for obesity. So 3 of the twin pairs were dizygotic and 1 of the twin pairs was monozygotic, and they were discordant. So 1 twin was lean and 1 twin was obese. And so you can see here that the principal components of the fecal samples clearly distinguish the lean human twin from the obese human twin. And then they took stool from the humans and they implanted it into gnotobiotic or germ-free mice. And when you take the fecal transplant from the lean twin and you put it into a gnotobiotic mouse, that mouse stays lean. But when you take the fecal transplant from the obese mouse and you from the obese human and you implant it into the gnotobiotic mouse, in fact, that mouse becomes obese. And this is demonstrated here in these curves here. So this mouse actually becomes obese. So it's really interesting to think about that the fecal matter in and of itself is sufficient to induce obesity. And what we find here in this huge heat map is that you get marked coordinate upregulation and dysregulation of branch chain amino acid pathways in coordination with development of obesity. And one interesting part of this study that I think is really interesting is that, you know, my husband says that I'm an eternal pessimist, right? I always think that bad prevails over good. So, you know, they put the 2 mice in a cage together. So they took the obese mouse and the lean mouse and they put it in a cage together. And I guess when mice are in cages together, they share fecal matter. So you would have thought that the lean mouse would become obese after sharing a cage with the obese mouse, again, because I'm the eternal pessimist. But in fact, the lean mouse was able to rescue the obese mouse and the obese mouse became lean. So just a little warning to all of you to be careful who you shake hands with. So, you know, I mentioned that the liver may be an important source of branch chain amino acids and determination of not just what your circulating branch chain amino acids are, but also why branch chain amino acids may be important in the biology around branch chain amino acids and the pathophysiology of branch chain amino acids. And I'll tell you, one major issue, I mostly, work on human cohorts, and one major issue in our human biomarker studies is what is the tissue source of the blood biomarkers? And this is particularly complicated in obesity studies where there's so many different organ systems that are, potentially involved: adipose tissue, liver, heart, vasculature. But I'll tell you, one likely source of circulating branched-chain amino acid is the liver. And this is work from my collaborators at the Duke Molecular Physiology Institute, Phil White, who is a very talented junior faculty, and Chris Newgard. And several years ago, there was a study from another group that showed that brain-derived insulin signaling inhibits liver branched-chain keto acid dehydrogenase. So as I told you before, branched-chain keto acid dehydrogenase is an enzyme. It's actually the first irreversible step in branched-chain amino acid catabolism in the mitochondria. And Phil and Chris recently showed that fructose feeding, such as what's seen in obesity, right? So excess feeding, excess sugar feeding, leads to phosphorylation. You get phosphorylation and increased expression of the inhibitory BCKDH kinase BDK, and decreased expression of the activating PPM1K, the phosphatase PPM1K. And so this leads to inhibition of hepatic BCKDH. And when you get inhibition of hepatic, hopefully you guys can see my arrow, when you get inhibition of hepatic BCKDH, this leads to accumulation of hepatic BCKA, and you get accumulation of fat in the liver, leading to NAFLD. And this suggests that this leads to de novo hepatic lipogenesis and increased fat storage in the liver. So it does suggest that the liver is an important organ for branched-chain amino acid catabolism in obesity, and may be an important source of branched-chain amino acids. So while we're measuring these branched-chain amino acids in the blood, and that they may be coming from adipose tissue, may be coming from the liver, and likely are coming from the liver. And in fact, this is human data that we have from liver biopsies from patients with obesity, where we looked at how branched-chain amino acid and branched-chain keto acid levels correlate with steatosis, role of branched-chain amino acids, branched-chain keto acids, and the role of this branched-chain keto acid dehydrogenase complex in steatosis. And what we have here on the left-hand side of the screen, on the y-axis, is the ratio between branched-chain keto acids and branched-chain amino acids, and along the x-axis, the steatosis grade. You can see this linear relationship with higher levels of branched-chain keto acids in higher steatosis grades, and higher expression of the inhibitory branched-chain keto acid dehydrogenase kinase (BCK) in the livers, suggesting again that this phosphorylation of this inhibitory kinase may be responsible for excess branched-chain amino acids. It also suggests a potential model. This is something that my lab is studying and many labs are studying, for crosstalk between the liver and the heart. So it may be that branched-chain amino acids are coming, this is a proposed model, branched-chain amino acids and branched-chain keto acids are actually coming from the liver and are having adverse effects on the heart. And while we're not going to be talking about heart failure today, we have lots of data to suggest the adverse cardiovascular consequences in the heart of branched-chain amino acids. And this actually may be adverse crosstalk between the liver and the heart. So while hopefully I've shown you that this is important biology that links obesity, branched-chain amino acids, and cardiometabolic disease, I'm in the end a cardiologist. I take care of patients, and I think what's really important is to think about biomarkers and things that we can use to better take care of patients with obesity, and to go back to the concept of this heterogeneity of not all patients with obesity go on to have events, and how can we better risk stratify and take care of patients with obesity, building upon these molecular insights. And so, in fact, our lab has shown over the past 10 years that these branched chain amino acids, these circulating branched chain amino acids, can serve as biomarkers in a variety of different contexts. This is work that we did several years ago that showed that branched chain amino acids may serve as better biomarkers for discrimination of metabolic wellness. So I showed you data from the NHANES study earlier in this talk, where they split up normal, overweight, and obese individuals by metabolic wellness. And we did the same here, where we split up lean, overweight, and obese individuals in a combination of different studies by metabolically well and metabolically unwell, with metabolic health classified clinically. And then what we did here, we then did metabolic profiling. What we found was that these branched chain amino acids, along the y-axis here is what we have is the branched chain amino acid factor levels. And while you find that, there's higher levels of metabolic unwellness in the obese individuals, as you would expect, that in fact, when you look at branched chain amino acid levels, you find that even amongst people who would be classified as metabolically well, for example, in the obese group, that they still have higher levels of branched chain amino acids than, for example, lean individuals who would be class acids may be useful as more granular biomarkers of somebody's metabolic health. Can you guys still hear me?
Yeah.
Okay, great, thank you. Just want to make sure. And in fact, what we've shown is, that suggests, those are suggestive data because the levels are higher, but now what we did in this study is actually tagged it to cardiovascular events. And what we have here is just different levels of AUC across, this is looking at a large cohort overall, and then stratified by lean, overweight, and obese individuals. And this is AUC curves looking at just metabolic health models described clinically. And then metabolic health model, the clinical model, with addition of a variety of different biomarkers, including branched chain amino acids in the green. And then this is metabolic health plus a variety of different biomarkers, including granular lipid biomarkers and branched chain amino acids. And in fact, when you add these molecular biomarkers to a more traditional metabolic health measures, you do a much better job of predicting who is going to have a cardiovascular event. And this is not just work from our lab, but this is work that other people have shown. This is work from Rob Gersten's group and Tommy Wang's group in the Framingham study that also shows that branched chain amino acids predict risk of future diabetes, in fact, up to 12 years in the future. So in patients who don't even have any evidence of insulin resistance, the higher your levels of branched chain amino acids, the higher your risk of having incident diabetes. And then how about heterogeneity and how we think about weight loss interventions? So those were biomarkers of sort of thinking about what somebody's metabolic health is and what their risk of cardiovascular events are. But we also know that we do a kind of a, I would say, not a great job of trying to figure out what somebody, what we should do about somebody's weight loss. What's the best intervention for somebody? And, there's really a huge disconnect between the amount of weight that somebody loses. This is another area of heterogeneity, and the improvement in metabolic health. This is data that we have from the weight loss maintenance clinical trial. This is a behavioral weight loss maintenance trial where they looked at different behavioral weight loss strategies. And this is plotting weight loss along the x-axis and along the y-axis change in HOMA. And while people lost weight, and you could say that weight loss is an important marker, in fact, weight loss doesn't necessarily very well predict how much of an improvement in cardiovascular mortality you're going to in fact, it's a pretty poor predictor of that. And in fact, what's probably more important is how much your metabolic health improves rather than how much weight you lose. But in fact, there's a disconnect between how much weight you lose and what your change in insulin resistance, for example, is. And, you know, here's this person who, you know, only lost, you know, a little bit of weight and had a really profound improvement in their insulin resistance. And then this poor person who lost a lot of weight and had a very little improvement in their insulin resistance. So how do we really, how can we use perhaps biomarkers in trying to dissect out this heterogeneity and who's going to have the best response in weight loss? And so we did the same thing, metabolomic profiling in some of our weight loss studies. And I promise you, it's not just that we only look at branched chain amino acids, but looking at a wide variety of metabolites. We again found that it was only these branched chain amino acids that measured at baseline before somebody has their weight loss interventions are able to identify people who are going to be the most responsive and are the most, these branched chain amino acids at baseline are the most predictive of improvements in insulin resistance to these weight loss interventions. So on the top hand side of the screen, these are data from the weight loss maintenance clinical trial. I just showed you data from the previous slide, that the higher your levels of branched chain amino acids at baseline, the greater your improvement in HOMA with the behavioral weight loss. And in fact, your branched chain amino acid levels change in parallel with changes in HOMA. And on the bottom slide, this is work that we did with Blandine Lafayette, where we showed that in patients who were matched for the amount of weight that they lost, but one had dietary weight loss intervention and one group had surgical weight loss, that branched chain amino acids decreased to a greater amount with surgical weight loss as compared to dietary weight loss. And this is in the Columbia cohort, this is in our Duke cohort, and suggesting that there's something about surgical weight loss, whether it's because of the microbiome, that has a greater effect on branched chain amino acid catabolism and homeostasis, and may explain the greater efficacy and long-term benefit of surgical weight loss. And this is work that we just completed with, in collaboration also with Blandine Lafayette, looking at large studies, the labs and look-ahead studies, where we found that branched chain amino acids measured before surgical and behavioral weight loss, as well as dietary precursors, you know, these are microbial metabolite precursors of TMAO, betaine and choline, are associated with who's going to have remission of their diabetes after weight loss. And you can see here that these metabolites predict who's going to have diabetes remission, and, you know, modestly, I would say, add to a clinical model for prediction of who's going to have diabetes remission. And then finally, since, you know, I'm a cardiologist, I'm interested in, well, what do these do for coronary artery disease and cardiovascular risk? And again, I promise you, we're not just looking at branched chain amino acids, but when we look at, you know, 100 targeted metabolites, we find that the branched chain amino acids are the ones that best discriminate patients with coronary artery disease and add incrementally to clinical risk factors. And then more recently, we've taken the view, the sort of technology around metabolomics has really evolved, and we've been able to take a more comprehensive view of the metabolome. So in the past, what we've done is really been very careful in using targeted, comprehensive metabolomic profiling, but with using spiked in internal standards. So we can do absolute quantitation, and we can be very sure that what we're measuring is what we're measuring. But the technologies have also evolved that now we can look more comprehensively across the metabolome using non-targeted metabolomics in a semi-quantitative approach. And this is work that we've done recently, taking a more comprehensive view of the metabolome, looking at surgical weight loss, behavioral weight loss, and exercise-induced weight loss. And what we find is when we take this more comprehensive view of the metabolome, that yes, the branched chain amino acids are significant in who is going to have the best metabolic consequences from a weight loss intervention. But in fact, what we find is there are metabolites that are much more significantly predictive. So these are metabolites measured at baseline before the weight loss intervention for who is going to have the best metabolic consequence, so the best percent change in HOMA with weight loss across this variety of different interventions. And in fact, these metabolites are diacylglycerols and triacylglycerols. So these are complex triglyceride related complex lipids. And even after you adjust for clinical variables, including triglycerides, these metabolites are highly predictive of who is going to have the best metabolic consequences from these diverse weight loss interventions. And this is work that's been led by a very talented computational postdoc in my lab, Nathan Billmeyer. And what he has also done, what Nathan has also done is to say, okay, well, those were metabolites that predicted who's going to have the best weight loss intervention commonly across these different weight loss interventions. But can we identify metabolites that are going to determine who is going to preferentially have the best weight loss intervention depending on what kind of weight loss intervention they have? So for example, would we be able to say, oh, you know, your levels of XYZ metabolite are higher, so you should have you know, a surgical weight loss intervention, or you have this particular metabolite, so you should have a behavioral weight loss. And so he did work where he looked at heterogeneity in metabolic, looked at heterogeneity of effects, and he found several metabolites that were associated with heterogeneity across these different weight loss interventions. So for example, this complex plasmalogen, which is involved in fatty acid metabolism, was to benefit from behavioral weight loss intervention, whereas this metabolite identified people who were not going to benefit from surgical weight loss. And then finally, we are doing some work we're doing some work with proteomics in the same weight loss intervention cohorts, where we're looking at proteins now that might predict who is going to have the best response to weight loss interventions. And we're finding a wide variety of proteins that also predict who's going to have the response to weight loss interventions. But in this case, in fact, we're finding proteins that predict who's going to have the worst response to weight loss interventions. And we're just starting to work through these data, finding a wide variety of different biological pathways, including cytokine pathways. And interestingly, our best biomarker, which predicts who's going to have the worst response to weight loss interventions, is actually NT-proBNP. So the higher your NT-proBNP levels,
Hopefully we'll get Svati back real soon. Hope everyone's well this morning. Just for color, she lives in the woods, and when I was still at Duke, she didn't even have, they had call-in access to the internet. I suspect it's evolved since then, but but maybe we'll give her a minute or two. Hopefully she'll get back on.
Can you guys hear me?
Yes, we can.
I am so sorry. I apologize. I live in the woods and my internet is crazy. All right.
I actually, it's funny, Svati, I just told the group that I don't know if it had changed since I was there, but I remember you had some challenges with your internet living in the woods as you do. So.
My husband forced me to live in the woods, so I apologize. It's beautiful out here. We live on 13 acres, but we don't have hardwired cable. So the quarantine has been particularly hard on us. I am so sorry.
That's okay. We don't have your slides up, just so you know.
Okay, great. Let me share those. Can you see them now?
Yes,
Yeah,
we can.
Awesome.
Great.
Okay, brave new world of coronavirus. All right, so just to finish up this piece, so I will tell you, I think one of the coolest things I've been able to do at Duke is to work with collaborators to think about implementation science. So this is work that we're doing now with the Translating Duke Health Project, working with Neha Pagadapati here, where we're actually using branched chain amino acids. We're doing home biospecimen collection, e-consent, sending people biospecimen collection kits, doing plasma blood spots, measuring branched chain amino acid levels, working with our BME colleagues then to create tissue engineered blood vessels from these blood specimens that we'll collect, and then using branched chain amino acid levels to basically stratify patients into behavioral interventions using mHealth. So just to kind of finish up, I want to say, look, my lab, I love doing biomarker work. I love thinking about molecular pathways, but to really tackle obesity, we're going to need way more than just biomarkers. So in the next couple of slides, I just want to tell you about some work that's being done across the country that I think is really impactful and really important. That really is going to, I think, have huge effects on obesity. So if you guys haven't heard this story, I really encourage you to watch this TED Talk. This is from the mayor of Oklahoma City. Oklahoma City, several years ago, the mayor was looking at U.S. News and World Report, which had ranked cities across the country for obesity. And in fact, Oklahoma City was ranked as the second most obese city in the country. And the mayor was completely horrified. And you can see here on the picture that he himself also was obese. He was over 200 pounds. And he was horrified by this. And he gives a really amazing TED Talk, if you ever get a chance to watch, where he talks about how he decided that he was going to have Oklahoma City lose 1 million pounds. And what they did was seemingly relatively simple. They didn't use biomarkers. They didn't do any of that crazy stuff. They used social media, healthy competition. He modeled behavior. He lost a lot of weight himself. They worked with local government. Obviously, he was able to since he was the mayor, where they changed the community infrastructure. They made the streets narrower. They had simple things like they made the stop walks, the lights go for longer so that people felt more comfortable walking in the streets. He built a big park in the middle of Oklahoma City. He made the city less car friendly and more people and bike friendly. And over the course of 2 or 3 years, Oklahoma City lost over 1 million pounds and went from being the second most obese city to being in the top 10 least obese cities. And I think this is just such an amazing story of, look, we're doing all this research on obesity and what we can do, at a molecular level, but these are really, relatively simple things that people can do working with city government to improve the health of our nation. And just to finish up on this piece, these are maps of childhood obesity, which just make me want to throw up even more than the adult obesity maps. And you can see that, again, the South is certainly more afflicted than the North here. And this is work that we are doing at Duke through an American Heart Association SFRN grant, where we are working with local government, the city government, and working with the park system here to do interventions with obese children and a variety of different interventions. And our program is called Hearts in Parks, and the clinical intervention is led by Sarah Armstrong. And we're doing some molecular work where we're collecting samples on the children and collecting fecal microbiome samples and doing fecal transplant studies to see if we can figure out, is there a set point of obesity and molecular obesity in children as compared to adults. But really what's amazing is how Sarah has been able to work with the local and city government to improve the health of obese children in Durham. I'm just going to skip that. So to wrap up, what do we need to do to solve this problem of obesity? What is the future? What may the future look like? Well, I think hopefully we'll have precision guided approaches where we'll have deeply phenotyped longitudinal cohorts. Hopefully we'll use integrated omics biomarkers that will be able to be done cheaply for more targeted interventions, so we don't have this sort of trial and error approach to how we take care of patients who have obesity. Hopefully we'll have decision support tools that are integrated into electronic health records, including digital health data and dynamic return of data to patients and people. Hopefully we'll have collaborations with city, state, and federal government, like Sarah's doing with our Hearts and Parks study, and as Oklahoma City did, community engagement, addressing disparities, and I think importantly, patient empowerment with behavioral and economics and having patients use their own data. And I wanted to mention Project Baseline because we are doing a lot of this in Project Baseline, not specifically for obesity, although obesity is a focus and cardiometabolic disease is a focus, but we are returning results to patients, participants in Project Baseline. We're empowering participants. It's deeply phenotyped, longitudinal cohort. And the reason I mention this is we are looking for collaborators, so feel free to email me if any of you are interested in working on any of the data from Project Baseline. We'd love to work with you. It's a huge amount of imaging, clinical, and molecular data. So what should we be doing for now in how cardiologists think about obesity? I think we need to screen annually. We need to promote healthy eating and exercise. We need to promote Life Simple 7. This doesn't take that long in clinic. We need to manage obesity as a long-term chronic disease with continual management, so not just a single conversation, but continual management. Screen for and manage cardiometabolic and other consequences, including LFT screening, screening for asthma, sleep apnea, and depression. Thinking, if you have a young woman, thinking about infertility and pregnancy counseling. And then I think as cardiologists, we're not so good at this, or at least we're not so good at this at Duke anyway, except for we have a small number of colleagues now that are really thinking about this, but we do need to consider more intensive weight loss interventions, including surgical and pharmacologic weight loss for patients who have severe obesity or who have severe metabolic consequences who have failed lifestyle interventions. So in conclusion, obesity is a major public health problem. Cardiologists need to pay attention to this. This has been proposed as the new field of cardiometabologists. So Neha Pagadapati here at Duke is doing this. I would propose that we need a more personalized approach to how we take care of patients with overweight and obesity. And I think in the future, hopefully biomarkers will play a role in that. We need to study overweight and obesity across the lifespan. It's really sad to think about the fact that children as young as 8 are having to have surgical bypass, but it is happening, and so we need to study it across the lifespan, and we need to address access to care and disparities. So I wanted to thank my entire lab, and I particularly wanted to thank all of you for putting up with all of my horrible electronic issues. And with that, I would be happy to take questions. Thank you so much for having me.
So, Dan, are you going to take the question, or I take the question?
You can.
Okay. So I don't see anybody's raising hand, or you guys can unmute yourself.
Maybe I can first start on behalf of the section, and can't be so, I'm so excited that you were able to join us, Svati. I do promise to show you the New Haven scenery someday, and we'll have to find a good excuse within the next year to bring you back for a different venue.
Be great.
And I can't, a little bit paternalistic, but I cannot tell you how proud I am to see how much how much knowledge you've created and how much you've advanced the field. It's really been a a pleasure to see this grow over the decades that we've been partnering together. So I guess the question I have for you, you didn't mention, but there is so much of focus, and we could have a political discussion about whether this is appropriate focus, but on bariatric surgery and options that obese patients now are flooded with. And I was wondering if you could speak to what you know about the metabolite composition pre-post bariatric, and is kind of bariatrics the experiment that you showed with the rats, where you kind of changed you give them a fecal transplant in a different way, I guess. So if I could ask you to speak to the bariatric surgery intersection.
Yeah, I mean, I think as you know, Eric, bariatric surgery has amazing short-term outcomes when you look at metabolic effects, and even some of the shorter-term cardiovascular outcomes are very profound with bariatric surgery. I think the problems with bariatric surgery, regardless of what kind of bariatric surgery you're talking about, are some of the recidivism issues that occur with bariatric surgery. So I think having these coordinated care plans, where it's not just you're getting your bariatric surgery and then you're being left on your own, where we do need these long-term plans for how we take care of patients even post-bariatric surgery, is really important. I know that's not what you're asking. In the work that we've been doing, where we've done molecular profiling in patients and microbiome profiling in patients with bariatric surgery, we have seen, again, really profound decreases in a lot of these bad metabolites, and in particular the branched-chain amino acid catabolic pathway, where we see really profound early decreases in branched-chain amino acids and branched-chain keto acids. And again, whether this explains some of the beneficial effects of gastric bypass surgery, it certainly is suggestive. And even again, after you adjust, in that one study that I showed you that we did with Blondine, even after you adjust for the amount of weight loss, so it's not just that people who have bariatric surgery lose more weight, that even if you match for the amount of weight that's lost, there's something about bariatric surgery that has a more salutatory effect on branched-chain amino acid catabolism. I suspect it's because of the microbiome, because we also see these profound changes in the microbiome with the bariatric surgery patients.
So Svati, now I had raised my hand for a question, and actually you're doing a very important job identifying novel risk factors. As you said, the genomics is not going to be all the answer, and you need to understand also epigenetics and also metabolomics. A few questions that I had for you. One, actually back to Eric's question, is that there is a correlation between the incretins and bariatric surgery and GLP-1 levels and other incretins, and that may, I didn't know if you actually studied the correlation between the branched amino acids and that. But the key question I had for you is that you show that there are, it causes adipogenesis and the hypogenesis, and you showed actually fatty acid synthase going up. But when I look at your transcription regulators of it, and I think you just measured only the mRNA, which is I saw it SRABP1. I wonder if you look at how these are linked. I mean, what transcription factor? Is that SRABP1? Is that SRABP protein levels? What is that actually changed by this? And again, once again, is that any relationship with the visceral fat, sub-Q fat? Because you talked about obesity, but can you describe that actually branched amino acids are actually more associated with subcutaneous fat versus visceral fat, etc.?
Those are all great questions. In terms of transcription factors, we still have a lot of work to do to determine that. We don't have the right data, at least I don't have the right data sets to really make those correlations. I think in some of the mouse data, we're able to look at some of the transcription factors. In the TAC model, we looked at KLF15, with Mukesh Jain, but that was more in the heart failure component as opposed to the obesity component of it. For the second component, to look at regional adiposity, we have not looked at the correlation of branched-chain amino acids and different sort of metabolic biomarkers for regional adiposity measures, but we're doing that right now in some of our CT data sets. So we're able to, with these chest CTs that we have, we're now correlating hepatic fat with epicardial fat and abdominal fat in the PROMIS data set and some of our other CT data sets. So we'll have some more data there.
And any relationship with the incretins, or?
We haven't looked. That's a great question. We should look in some of our data sets. We have not done that. And we should look in some of our human cohorts.
I mean, Jeff was the second person to ask question. Jeff?
Yes, Dr. Shah, that's a terrific talk and terrific work. So Arya just asked about interleukins. That was going to be my question when you, in the gnotobiotic mice experiments with fecal transplant, whenever you see microbiome being correlating with or being a mediator, if you will, it always raises the question about differences in immune responses. And so that I was going to ask whether you looked at any inflammasome feature differences. That's obviously a very hot topic right now with susceptibility to severe COVID disease, which we think that may be an indicator. But have you looked at that at all?
We haven't. The fecal transplant study that I presented was from Jeff Gordon's lab, so I wasn't involved in that. We are looking at that in the pediatric study that we're doing. We haven't started the fecal transplant studies yet, but we will be looking at that in the pediatric obesity study that we're doing.
Thank you.
Larry, you're the next. Larry on?
Yeah, just unmute myself here. Thank you. That was a great talk. I was thinking back to the liver and and to what extent you mentioned central insulin signaling is maybe the trigger for all of this. I wondered if you could expound upon that. That's a very interesting concept. And then to get back at hepatic steatosis as either also a driver or a consequence of the branched-chain keto acids.
Yeah, I mean, I think this is evolving data. That one study from a few years ago, I can't remember whose lab it came from, suggesting that, brain-derived insulin signaling was sending a signal to the liver and was affecting BCKDH, enzyme activity is really fascinating. Yeah, so our data, when I say our, this is not my data, that was Phil and Chris Newgard's data suggesting that phosphorylation of the BCKDH phosphatase was affecting, branched-chain amino acid and branched-chain keto acid accumulation in the liver and was then, causing de novo lipogenesis in the liver. That's data that they just generated over the past year. Is really interesting data. What we're doing now is then trying to determine, this is some work that's being done by Rob Magera, who's a collaborator at the DMPI, is trying to understand how that's affecting cardiac signaling and heart failure in some mouse models that he has. But it's interesting data. What we're doing on the human side is taking a lot of our NAFL data sets and trying to understand now, looking at some of the liver biopsies that we have and understand branched-chain keto acid levels in those patients and how branched-chain keto acid levels are then affecting downstream risk of heart failure.
Margaret Furman?
Hi. Can everyone hear me? Svati, that was an excellent talk. My question goes back to a bit what Dr. Glasgow asked. When you commented about the benefits of the gastric bypass, I was wondering how far out were the studies carried that we know unfortunately many people do gain some of the weight back, but do some of the benefits linger beyond the fact that they gain the weight back?
I couldn't completely hear what you were saying. I think the question was, how far out do you see benefits from bypass surgery?
No, my question was more that we know many people gain weight back. Unfortunately, not everyone are successful at keeping the weight off. I was wondering some of the original benefits of the weight loss surgery, do they carry on even if they gain the weight back with the branch chains?
Meaning, do branch chains predict who's going to have recidivism in terms of weight regain?
Yes, but if they gain the weight back, do they still retain some of the benefit of the surgery?
Oh, yes. Yeah. The clinical data, those are not my studies, but yeah, if you look in the lab study, you do still retain some of the cardiometabolic benefits of the surgical weight loss, even if you regain some of the weight back. Yes. That's what the clinical data show from the large gastric bypass studies. Is that what you were asking?
Yeah. Well,
Yes. Yes. Yes, you do still retain some of the original benefits of having lost that weight. Yes, it's a great question.
Okay, Carsten?
Hi, can you hear me?
Yes.
Yeah.
Thank you for the talk. My question is, a little more simple. So, how about the dietary supplementation of, BCAAs? It's a lot on the market. What's the relationship between that and what you measure? So people that use, BCAA as a supplement.
Right. I mean, it's, they're essential amino acids. So theoretically, it's the same as, what you would be eating. So I would argue that those are not good. That excess branched chain amino acids, the same that you would get at, what's the name of that store that sells all those supplements, that they're probably not a good thing. So even if they're putting muscle weight on, that they're probably having adverse metabolic effects. I don't know if there have been studies of, weightlifters who take, branched chain amino acid supplements to see what the adverse metabolic effects are, but we certainly know that a lot of those supplements do have adverse metabolic effects. I think it's a good question.
Okay.
It's interesting. The one thing I will say, which we haven't been able to figure out from the human data, is pretty consistently across our data sets and other people's data sets, when you look at incident cardiovascular, if you look at incident heart failure, branched chain amino acids, the higher your branched chain amino acid levels, the higher your risk of incident heart failure. But when you look at all-cause incident coronary artery disease, higher is worse. But when you look at all-cause mortality, higher branched chain amino acid levels paradoxically are associated with lower risk of all-cause mortality. So the lower your branched chain amino acid levels, the worse your all-cause mortality. And that's sort of this weird paradoxical relationship that many people have seen in their data sets, which we don't completely understand. So anyway, I don't know if that's related to, some kind of homeostasis effect with how much branched chain amino acids you need. We also know that patients who have really bad liver disease have very low circulating levels of branched chain amino acids. So whether it's reflecting, that you're sort of end stage and you can't, it's sort of reflecting, low levels of protein. I'm not sure.
Thank you.
Okay, I don't see any further question if I'm not missing anyone. If not, I want to thank Dr. Shah to attend and give us a very terrific lecture. And so we learned a lot from you. And I'm sure many of us are actually interested in collaborating with you in certain areas of interest. And I'm certainly myself going to actually contact you. And, there are in obesity and metabolism, many of us study here at CVRC and elsewhere. So thank you very much, and I hope we see you here in person when this pandemic is over. Thank you very much.
Thanks, Svati.